Please take your seats. We are continuing with the rest of the program, and it's a great honor and pleasure to introduce Professor Geert Litjens to you. He is professor here at Nijmegen Radboud University Medical Center, and he actually bridges the field of radiology imaging with the field of pathological imaging. And well, I understood from him that today he will also bridge to the omics field, and perhaps still playing a second fiddle in his research, but maybe that will change after today. And we are very much looking forward to an interactive and also exciting lecture. And the fact that your team also deserved and won the Modo Award already signifies that you're definitely able to entertain this audience. So please take the floor.
Thank you, Peter Bam. I think my microphone is working, so great. Yeah, so I come from an imaging background, but like Peter Bam said today, I'll also try to bridge a bit to the cross-omics field. Yeah, nowadays when I give lectures, I don't really have to introduce the concept of AI anymore. I think most people here will have used it in some way or form, either for generating nice images of the Pope in a bomberjack, or if you have one of the latest Samsung telephones, you can actually do live translation of your phone conversations from and to different languages. So actually we predate the Star Trek Universal Translator by roughly 125 years. You can use it as a writing aid to help write your papers. If you do, I would recommend checking the text that is generated before you try to publish. Or you can use it, of course, to generate nice puppy videos nowadays. And I often also get the question, okay, what do you then use AI most for as a sort of professor in AI in imaging? And I always say I use it mainly to generate coloring pages for my kids. Now you might wonder, why do you need to do that? You can buy these coloring books. Yeah, but my kids have very specific wishes. They want coloring pages which have like Super Mario, Elsa, unicorn, all on the same coloring page. And then you can also see that AI is not perfect yet, because Olaf actually doesn't look very healthy here. But yeah, I mean, this is all general computer vision, right? So why would you ever want to use AI in something as deeply human as healthcare? Well, there are two main reasons. One is also one that is often mentioned by our government, and that's also based on this report. It's in Dutch, but it says, choosing for sustainable healthcare. And they sort of show this insight that our healthcare costs are ballooning relative to the rest of our economy, and that if we want to keep healthcare at the same level as we have it today, that now roughly 15% of the Dutch population works in some capacity in healthcare. In 2060, we will need 30% of the Dutch population working in healthcare, and that's simply not sustainable. This is not just a Dutch problem, obviously. This is something that you see all across the world, from the UK to the US to China, Japan. The same everywhere. And like I mentioned, our government realizes this. This was a letter that our then Minister of Health sent to our parliament, where he says, okay, it's very important that we use AI to make our healthcare sustainable and good quality. But I think sort of this focus on efficiency is only one part of the puzzle. And I think there's actually another area where AI can help us. Because when my grandparents used to go to the GP or to the hospital, it was very simple, right? So they would do some measurements, maybe blood pressure, BMI, at most maybe run an X-ray. But yeah, nowadays, and I think this community can emphasize with that, is that we have so many options of data that we can collect for each individual patient. And whereas all those years ago, you might get 1 or 2 pills or surgery, nowadays we also have so many treatment options, from immunotherapy to different kinds of chemo and radiation. So as a physician, it's not just that your work pressure is increasing, that you have to do more patients and more care. It's also that the care you have to give is getting more and more complicated. So I can imagine that sometimes you don't know what to do anymore. So how can AI help in these endeavors? Well, I usually use this very, very simple scheme. So for most decisions in healthcare, this is what happens, right? You get some data from your patient, like an X-ray or a CT scan. You extract some features from it, like is there a nodule present, and if so, how big is that nodule? and then based on that, you decide on a diagnosis or a treatment. And this is what medical doctors do every day, and for that they use still relatively simple population-level risk models. So for example, this is the one for prostate cancer, where you have clinical stage, you have a blood test, which is PSA, and you have a Gleason score, which is assigned by a pathologist after microscopic inspection of tissue. But yeah, as you realize, there are like multiple versions of omics, other imaging modalities, measurements that you could have added to this risk model to make it more personalized and more tailored for an individual patient instead of just operating at the population level. And I've tried to share already that like incorporating all of this information is just difficult for medical doctors. So why not have the computer take part of this diagram, right? So maybe we decide the data and the features, and the computer decides what then comes out. And this is definitely not a new idea at all. In radiology, already in 1964, we had the first AI application. And what they wanted to do was they would want to make an electronic scanner computer to look at chest photofluorograms and separate the normal from the abnormal. I can tell you, even today, we haven't solved this yet, but I mean, the idea was already there. And just to stress how long ago that was, people of my age and older still remember these things, right? Well, this was before that, because they had to feed in the data with these punch cards. So there weren't even floppy disks. So from those types of AI systems, how would we build them? Well, I'm just going to take a very simple application. That was the first application we actually worked on also with modern AI techniques, and that's the identification of cancer metastases in lymph nodes. So if you know the TNM staging, tumor, nodes, metastases, the lymph nodes are actually a very important determinant on how you treat a patient, because if the cancer is metastasized to lymph nodes, it's no longer contained to the organ, so simple surgery is typically no longer enough. And the way we assess metastases in lymph nodes is often through imaging, but they're always confirmed by a pathologist upon microscopic inspection. And this is a typical task that is ideally suited for AI. It's not mentally challenging, it's not difficult for a pathologist. Pathologists can easily do it. It's just that it's looking for a needle in a haystack. So it's very time-consuming and labor-intensive. So I have a short video to show for people. I think most of you will be familiar with histopathology images, right? But this is what they have to do. They go through all of these cells. So every purple dot is a cell nucleus. And they have to try to find the group of cell that is originating from a cancer. And just to help you a bit along, so in this slide, this is the entire metastases that they have to find. So you can imagine it's not a very nice task. So how would you build AI systems in such context? Well, very simply, for the old-fashioned one, where we would still do the feature extraction ourselves, as you would sort of sit together with a pathologist, you would look at normal cells, you would look at cancer cells, and you would come up with features that separate these two images. Which, well, very naively, if you look at it, the top is darker than the bottom, right? And it seems that the top, in this case, has a higher cellular density than the bottom. So let's assume that these two features would be good enough to do this classification. So you can then plot them very nicely in a graph, and I could just draw a line here, do a logistic regression or something, to separate these two groups. And why stay at two features? Why not try as many features as we can? And that's where sort of the field of radiomics and pathomics got started, because we also wanted omics, because it's cool and it makes it easier to get published, so why not? And we even steal your graphs and your figures, right? So we make it look very similar. But the idea here is that, well, you can calculate thousands of features based on these images, and if you plot them like this, you can see that you have features on the y-axis and patients on the x-axis, and then you can see that there's separation between patients based on these features, and you can do survival prediction with those types of features, for example. And this was reasonably successful, these approaches. There were actually commercial products on the market. This was developed by my PhD supervisor in the 1990s, 1995. This machine was developed actually before radiology became digital, so you had to actually scan the physical photographs to get them into this big computer, and then the AI would help you analyzing mammograms, so breast photographs. And that was also what I worked on in my PhD. This was my very beautiful thesis. And this was the result of the AI system I built. That's the blue line, and then the radiologist was on the green line. And this also immediately highlights the problem, right? So I spent 4 years doing my best, getting a good AI system, and then I still couldn't get anywhere close to the radiologist. And that's always why, if you talked about AI back in those days, it was always, okay, maybe as a second observer, so to help the radiologist when he's very tired on Friday and then misses something, then maybe that's the only situation that AI can be helpful. So why was it difficult? Well, there are two options. Either I'm a very bad AI researcher and I build crappy systems. In that case, you're in the wrong room, I would guess. Or there was something notoriously difficult about the way we were approaching these things. So I showed you this example, right? But yeah, as you already realize, often it's not that easy. Often it looks like this. And then coming up with the features to determine what is normal and what is cancer is actually very complicated. And you also have the translation layer of the pathologist telling you what they're looking at, and then you trying to program it into a computer. That causes performance loss. So how do we go from there? Well, in 2012, sort of the world of this way of doing AI ended. And it ended with an example from general computer vision, where we had models that could classify pictures in different categories, right? Like if there's an animal or a car or an airplane or a boat or whatever. And up to 2011, computers were also very bad at that task. But then in 1 year, we halved the error rate, and then now, well, we're at human level performance. And just to sketch a bit how impactful that work was, that paper, I think this is, I updated it a couple of weeks ago. It's cited 151,000 times. That's for every day since this paper has been released, 1 citation per hour, 24/7 for every day, for every year since this paper has been published. So in terms of scientific impact, I think it's there. So what was the key thing that they did different in this approach? Well, actually, pretty straightforward. So they said, okay, if the computer can do this prediction decently well, why not let it also come up with the features instead of us saying what the features are? And for that, they used deep neural networks. And actually, the concept of a deep neural network sounds very fancy, but it's actually pretty simple. So you start with an artificial neuron that gets certain inputs, can be whatever, can be omics, can be imaging, ECG, whatever you want. And then this is the only equation that you actually need for deep neural networks. It's a simple regression function. Because the only thing this neuron does is it multiplies the input with a set of learned weights, adds a bias, and then applies a nonlinearity. That's not even learned, that's chosen. The trick is, of course, that you don't just use one neuron, you use many neurons that are all connected to each other. And then this animation is probably a bit slow. And then, of course, you use more layers and more neurons, and then you can go to more layers and more neurons. And in the end, you can do some reasonable prediction, normal or cancer. And to give you a guess, this is, in this example that I draw here, is roughly 10,000 parameters, this neural network. If you look at the latest version of ChatGPT, it has 4 billion parameters. So this might seem like a very big neural network, but it's very, very tiny compared to what is out there already today. So and it was not just the neural networks, but also the way we then optimize these machine learning models. And I always like this example. So we don't program these AI systems anymore, we train them like dogs. And I think that comes closest to what we actually do nowadays. So to go back to this lymph node example, right? So there was this metastases here. So the only thing that I do now is I just extract samples of normal tissue, I extract samples of tumor cells, I give them to this deep neural network, and I say, okay, optimize your parameters such that you reduce the errors you make on these patches, and you figure out yourself what you need from these images to make that decision. And then you do that million upon million upon million upon millions of times, and then after this whole training process, you get an algorithm that can do this relatively well. So to sketch a bit, so I of course, when I started working with these deep neural networks after my PhD, I started comparing them to my PhD system. So my AI system had a performance of 87 accuracy. It was roughly 12 months of work for that iteration. And then 2 weeks after starting with these deep neural networks, I had a system that was significantly better. And I realized that my thesis contained a lot of content that would be very useful for a campfire. But it's also super exciting because at that time in our field, we started applying this to everything. So in those years, we were outperforming systems we've been working on for 20 years, like in a matter of months. And these are still, if you look at, not that citations are that important, but if you look at my most cited papers, it's all of these from the early times of this AI implementation. So it's also a very exciting time to be in the lab. Okay, so to go back to this lymph node example, so the reason I use this is because it's like very low-hanging fruit, right? So I said, okay, this is a task, it's simple for pathologists, it's just time-consuming. So the first thing you want to optimize. So when we started applying these AI methods to this problem, we had this is a duplicated slide. Let me just click over this. So we actually had a system that worked reasonably well, but obviously the chance that we as a single group at Radboudumc in the Netherlands make the best possible system is pretty small. And I'll refer to this idea a couple of times through this presentation. So what we did is we took all our data that we had, it was 3 terabytes of data, and we made it publicly available to everybody in the community who wanted to try to solve this problem. So in total was 1,500 slides, or 1,400 slides from 1,400 patients. And then we challenged the world to do this better than we did. And of course, everybody did. Well, not everybody, but a significant number of people actually did better than we did. And we actually compared those top-performing solutions, which the most actually came from Boston, from Harvard and MIT. And we compared them to a group of pathologists. And actually, these top-performing algorithms were the first time that we could show that these algorithms could actually do better than pathologists at a task that they can do relatively well. So, like I said, this was low-hanging fruit, right? It's a task that pathologists could already do. So we said, okay, let's take it a step further. So now we're going to look at a task that is also relatively difficult for pathologists, where they disagree a lot. And that's grading of disease, right? So you don't detect the disease, you actually have to say how aggressive is this disease and what kind of impact would that have on the survival of the patient. And use prostate cancer here as an example. The way that works is you have a suspicion, biopsy is taken, and then a pathologist assigns a Gleason grade that ranges from 3 to 5 nowadays, where 3 is low risk, and it's very similar to benign tissue. 5 is significantly higher risk, where you see that the cells start to lose their normal function. The thing is that this whole grading system is nice, but it's very inconsistent. So this is a study from Rotterdam among 35,000 patients, where they looked at the percentage of low risk that is at each hospital. So each dot is a hospital, and the x-axis is the number of patients they see a year. So what you see here, this is the average in the Netherlands, and this is the confidence interval based on variations in population between the different hospitals that you would expect. But if you are now at this hospital, you have an a priori chance of getting a low-risk diagnosis of 20%, and at this hospital of almost 50%. And it's not explainable by the differences in population, so it must be the differences in pathologists between those hospitals. And that's a problem, because here you will most likely not get treated or get conservative treatment, whereas here the chance is much higher that you get aggressive treatment, surgery, or maybe even worse. So again, we built an AI system exactly in the same way as I showed you before that could do this grading automatically, which was nice, and it could show, hey, there is this area of 15% that's a bit lower risk, but majority here is very, very high risk. And we again compare this to the pathologists. So here you have the inexperienced pathologists with less than 15 years of experience. And the dark blue dots are the experienced pathologists, so you can immediately see that as a patient, you want the experienced pathologist to look at your biopsies and not this one. So of course we anonymized all the pathologists in this study, so don't worry. And the average pathologist had an agreement with each other of 0.82. And the AI system that we built had an agreement of 0.85. So right in the middle of the experienced ones, but better than all the inexperienced ones. And that was very nice, but I also want to stress here, so this AI system is nice, but actually if you look at the entire study, the AI system is actually the least important part. Actually, the majority of the work is done by the PhDs and the 1,200 patients who were willing to give their data for this, and the 18 pathologists who allowed us to compare them to an AI system. And again, so we now have a different animal. We went from chameleon to panda. We did the same trick. So we took all the data that we used for this study together with the Karolinska Institute and made this available to the whole world. There was roughly 10,000 biopsies from 6,000 patients, and again challenged the world to do better than we did. And we had 1,000 teams participating in this study, where it's 335 shared solutions. This is the histogram of the top 100, and if you remember, we had 0.85, so we're here. And this was actually the top 100 of teams. There were 100 teams worldwide that came up with a better solution than we did. It was not just that they had a solution for this data set that we provided, we also took those top solutions and compared them on further data sets from Karolinska Hospital and from Google Health, so from the US and from a different hospital from Sweden. And we have the algorithms here. And we also compared them to the pathologists from the US and from Europe. I see that the algorithms actually are also for this, where we thought, okay, this is a difficult task, even for pathologists, the algorithms are doing a better job. So, where at the time of my PhD we were here, so we're talking about, okay, maybe if the pathologist has a very crappy day, the AI can help, to a situation where, well, at least you can say they're sort of equal in performance now, right? And there are also a lot of commercial products around that now. Okay, so I've been talking a lot about imaging up to this point. So where is the omics, right? Well, actually, a lot of the techniques that we were using here didn't really work very well for omics type of data. And not just for omics, but also, for example, for text. So why was that? Well, one of the reasons, so not the only reason, but one of the reasons is that these models, which were typically convolutional neural networks, were very bad at capturing, for example, long-range dependency. So if you have this sentence, the report that the assistant to the manager who had been promoted after the unexpected resignation of the previous director of operations submitted last Friday shocked the entire board of directors. To understand what this report did, you have to find this word. And that's something that these models were at that time very bad at. So that changed in 2018 when you have this cool action figure. Well, the action figure is not important, but there was a paper from Google that was titled "Attention is all you need," and what they introduced was what they called the transformer model, which is why this picture is there. And they used actually two key insights. So they introduced a self-attention mechanism, and I will not explain it in detail because that gets a bit too complicated, but the idea is that it would calculate for each element in a sequence the importance of all other elements in that sequence to understand that word. So to understand it, animal was the most important one, and less than street, less than it. And in a different sentence, it was more important for relative to street. And the second, so that allows these models to capture long-range dependencies. Of course, it's a bit more complicated than that, but it's the general idea. And the second aspect they introduced was unsupervised pre-training. And this is for a language model, so the GPTs ones that you know very well. And the way they work is you just take a piece of text, you give that to the model, and then you say to the model, predict the next element in this sequence. So in this case, the next word in this sentence. So if that's correct, you say, good job, model. If it's bad, you say, bad job, model. And then you just take that output, you add it to the input, and you do the same step again. So what is then the next word after this? And then the next word, and then the next word. And then you just train it on the internet, and then you end up with ChatGPT. But of course, this is not just relevant for language, this is relevant for anything that can be expressed as a sequence. So also for genomics, or for amino acid sequences, or other aspects where you use sequences. And well, I think most of you will know the Nobel Prize last year went to a group of scientists from DeepMind that actually applied these types of techniques to predicting the 3D structure of proteins from an amino acid sequence. And this is how that model looked. I will not explain it in too much detail, but they combined multi-structure alignment, so they aligned sequences of amino acid across species, because that gives you information if, for example, amino acids in a sequence jointly change position, that might have implications for the structure, right? So you can leverage that. And you, of course, have existing 3D structures for which you know the location of the atoms and the orientation. And then you actually put that into this transformer. So this transformer has 48 blocks. That's not super important, but it's a very large model. And then you can iteratively use this to optimize for a specific 3D structure. And so this won then the Nobel Prize. They have made various iterations of this model by now. But that sort of was a wake-up call to other AI researchers in the field, like, hey, this field of omics now starts to become viable for our AI techniques. So people have followed up. So this is, for example, a publication from Meta, from Facebook, and other things. So this is actually a relevant development in the world. And they showed that, okay, but now you can just take any language model and learn it sort of the protein language or the genomic language, and then leverage that for science. And this was a publication in Science last year, where this was the first DNA foundation model. So this was a ChatGPT-like model that was trained on the genetic code of 80,000 bacteria. I think it was around 800 billion nucleotides in total that this was trained on, in exactly the same way that ChatGPT works. So you take, sorry, wrong button. You take your nucleotide sequence and you ask it, okay, predict the next nucleotide in the sequence based on all this real-world genetic material that you have. And then you can generate, so if you want to try it out, you can find the code for this. It's called EVO. It can generate genetic sequences of up to 650,000 nucleotide pairs. And what they did is, okay, that might not sound interesting, but what they show is that you can generate these pairs, and then you can take the likelihood that this pair is feasible from the model itself, and you can compare it to things like fitness from experimental studies. So how likely is it that this sequence would survive in nature? And that way you can actually probe for a lot of different sequences. Okay, how likely is it that I have this sequence and that it actually would occur or be feasible in the real world? And in that paper, they actually show a lot of different other applications, from RNA to protein structure prediction as well. So if you're interested in this type of thing, I highly recommend reading it. They actually have an updated version, EVO 2, that is on bioRxiv, so that might also be a relevant read. Okay, so this is what we can do today. So where should we go in the future? And I'll talk about that in 3 different directions. So right now, everything I've been talking about is something that humans can also already do, right? So for example, for the pathology part, detection, grading, that's something that humans can also do. So I think we should also start exploring options of applying AI to things that humans cannot yet do. So how would that potentially work? Well, I'll take the prostate example again. Here what we did up till now was just saying, okay, this is the reference standard, right? The Gleason grading done by a human, just replicate that reference standard. That was the goal for the model. But what now if we say, okay, but just skip all that. You just take the survival of the patient and directly predict from your image data or from your omics data or whatever, whether the patient will survive or not. So that's actually something that we did last year. So we had samples from prostate cancer patients, and we had to tell the model to predict the years to progression, specifically biochemical recurrence. It was actually very nice that the model was actually able to do that. So we could separate, we validated that on 2 cohorts from the US, could separate patients with low, short and long-term survival. And you already feel maybe where this is going. So we took another animal, made all that data public, 2,000 patients from 4 different sites. And we asked people, okay, do a better job than us. So predict recurrence, so biochemical recurrence in the case of prostate cancer. We had binary labels, so does this recurrence happen within or within 5 years or after 5 years, and we used a C-index. So we first took, okay, how well do the pathologists do, right, for this survival prediction? So pathologists get a C-index around 0.7, 0.72, 0.71 in predicting, okay, this is the tissue, will this patient get recurrence or not? And the algorithms independently, so without using Gleason grading, roughly same ballpark, so there was statistically no inferiority of the algorithms to the pathologist. But if you actually combine these two, you do better than each of them individually. So actually these algorithms learn something from this data that the pathologist did not yet know, or do not yet know. And that's of course interesting, right? But yeah, the problem is these AI algorithms are a black box, so we don't actually know what they are using. So that's something that we started looking at. And yeah, if you look at literature in this field, what you get a lot is sort of this, right? So you could highlight in an image with attention maps, you could highlight which areas are relevant for their decision. If you take this to a doctor, there's nothing they can do with this, right? So what is then in this red area, what is relevant to this model? Is it the size of the nuclei or of the cells, of how they are ordered? It doesn't tell you. So we took a slightly different approach. We took prototype images and we clustered them actually based on their association with the recurrence. So it might sound all complicated, but these are patterns that according to the model are associated with soon, quick recurrence, and these are patterns that are associated with long recurrence. And we went to the pathologist with that, and they found these choices reasonable. There's things that they could also see. But actually, researchers from Google took this approach that we developed and then used it for colorectal cancer. What they did is they took this top feature for colorectal cancer. This was in colorectal cancer sort of, you can describe it as fat invasion. And they taught pathologists to score this AI-identified feature. And what was very nice is that they showed that if pathologists score this feature in 3 different, like category 1, 2, and 3, that this very nicely separated colorectal cancer patients based on survival. So this was the first example of a biomarker in pathology discovered by AI that humans could then use to improve their prediction. So I think we should be doing more of that. Okay, from narrow to broad AI, I think this I can explain very simply. So all of these applications I've been talking to you about right now are narrow AI. So you make an AI model that does one task well, but cannot do anything except that task, right? The thing is that doesn't scale. We have so many diseases, we have so many applications in the clinic, you cannot make an AI algorithm for every single application that you would want. So what we would actually want is, I would call it broad AI because I think general AI is the wrong term. We're not anywhere close to general artificial intelligence. And for that, you have foundation models, or we're developing foundation models. So they are called foundation models because they're intended to be built upon. So foundation model by itself doesn't do a lot. It just lays there and then you build actual relevant things on top of that. And for a long term, you had ChatGPT and things that were relevant for computer vision, but also in the pathology field now we have foundation models that are trained on millions upon millions of slides from patients in an unsupervised fashion, and these are all their names. There's a new pathology foundation model every week. But where they are relevant is areas where you don't have a lot of data. So all of these applications I've been talking about, prostate cancer, breast cancer, we have lots of patients, we have lots of data. It's very easy, is not maybe the right word, but it's very straightforward to build good models for that. And if you're suddenly in a cancer domain, for example, or a disease domain where you don't have a lot of data, these foundation models can help a lot. And to show you an example, this is a publication from Faisal Mahmood's group in Harvard in Nature last year, where they discussed their foundation model. And they show here, of course, they test it on a lot of common cancers, right? Breast, prostate, lung, colon, and it works really well. The foundation model Virchow, performance is above 90%. But the actual interesting thing is that also with very little data, sometimes only a handful of cases, below 10, they also could get decent performance on very rare cancer, or very rarer cancers, so like stomach cancer or liver cancer. We're not there yet, because if you compare the performances of these broad AI, so again, for example, on Gleason grading, let me just click through the animation. So if you look at broad AI, so these foundation models, they have a decent performance, actually better than I would have expected by now, but they're not yet anywhere close to narrow AI. So they're useful, but not good enough yet. The last one, which I think also for this community might be the most interesting one, is we need to go from unimodal approaches, so from pathology or radiology, to multimodal approaches, so multi-omics or multi-data, whatever you want to call it. And I think the main reason for that is that we tend to look at these patients in isolation, right? So I have a prostate cancer patient, I'm a pathology, so look at the pathology of a prostate cancer patient. But for this patient, it's not just the pathology. They come in, they get a blood test, or there's a potential for AI, they get imaging, they get biopsies, it's Gleason graded. If it's not too severe, they might get active surveillance with recurring follow-up scans. They might get laser ablation treatment. They might get surgery if it's a bit more severe, or radiation therapy, which at Radboud we nowadays also do under MR guidance. Maybe, sadly, they might get distant metastases. We also do imaging for that. At a certain point, you start collecting omics data for these patients that might infer which radiotherapy or which chemotherapy you want to do. Or you do molecular treatments like lutetium PSMA. And the thing here is that you have all this wealth of data for these patients, and here you have a lot of patients, right? So this stage. Diagnosis, you have thousands of patients. But once you're here, it's only tens of patients that are left. So we should use all this data that we have in front here to help AI models make these decisions as well. And also, this is not a unique idea. There are people working on this. For example, Google has a model they call MedPalm. They have a bit of a fancy title towards generalist biomedical AI, also why I don't like the generalist term. And what they say, okay, we have models for all of this data. So we have a pathology foundation model, radiology foundation model, genomics foundation model. And we use all of that data, we learn these foundation models, and then we freeze them, and then we make a prediction model. But that's not the way the world works. So just to compare that to the real medical domain, so we have a radiologist and a urologist and a pathologist. We train them for 6 years, they specialize, and then we would say, okay, we now put you in ice, and you cannot learn anything anymore, right? So you cannot learn from your colleagues. Whereas in the real world, they are always a team, right? So they learn from each other, they improve together. And yeah, if I have a pathologist that sees all these false positives come by in his biopsies, they will tell the radiologist, hey, you need to do a better job, or what you're looking at in your image is not the right thing. So how should it work? Yeah, I need to hurry up, I noticed. So how should it work? Well, this is the old-fashioned way, right? So you would have 2 different models that extract features. You would have sort of this mixing layer that would allow you to predict, for example, biochemical recurrence. And I always make this split at radiology pathology because this is where you connect the in vivo and the ex vivo world of the patient. And of course, here you have omics that can be added to this. Here you have clinical information. But ideally, you would be able to also fuse these methods. And I think this is the last example I have of people who are doing that. It's from, again, Harvard, from Chen et al., Cancer Cell 2022, where they combine genomics data and histology data. So the way they do this is they combine this foundation model for histology imaging, but they also have RNA expression, copy number variations, and mutation status for different genes. And they combine the histology and the omics features and then train hold this thing end to end. And they use this explanation to predict patient survival. And they also use explanation, so which gene or which copy number variation is relevant for this survival prediction. And they also use these attention maps. And what they show is that if you combine radiology and pathology and omics data, that you, in all cases except one, I think, actually outperform the single one. I'll wrap up quickly. So how do we move forward? So one thing, and that's I think also relevant for the omics field and also what cross-omics is about, is that we should get away from the situation where we think that all the data, my own data is mine, and we should not share things. And I think I've shown you a zoo of animals, of challenges, where we try to get that done. So I would click through this. I would recommend that you look at the Grand Challenge platform where we host all these challenges, because we're also expanding it now for omics, so you can also run omics challenges on there. And actually this year we have the first challenge where we have paired H&E and RNA sequencing data for bladder cancer patients that we are running. So that might be of interest to you. And that might eventually get us to an AI model that can incorporate all of these different types of data. I'd like to acknowledge all the people in my group who do the hard work and allow me to take credit for that. And if you have any questions, I'd be happy to answer them. Thank you.
Well, thank you for this very educational talk. The audience may come up with questions, but let me ask one first. So when you think about the future and the practical implications of this, you have some experience in combining computational pathology with medical imaging from the radiology and building a workflow around this. Say you have to add omics to that as well. What is the main practical implication of that, and how would you do this?
Yeah, I think it depends a bit on which type of omics data. So for example, for spatial transcriptomics, I actually think you can treat it in much the same way as other imaging data, but just your feature dimensionality is much larger, whereas your resolution might be lower. If you look at other types of omics data, what we've done in the past, or essentially the main way we do it right now, is you first, it's essentially a model architecture question. What we now typically do is either you have a spatial alignment between the imaging modalities, you combine them, you have one feature extraction pipeline, and then you add omics features to the end of that, and then you can train it end to end. Because yeah, you don't know, there's no spatial relationship between the omics data you have and the pathology data. Yeah, I think that's the standard way to do it. So it's mainly an architectural design thing.
Yeah, we saw this morning indeed that you can, in fact, create a kind of a copy of your slide and use omics profiling of that. So that may be one of the future directions. Are there questions from the audience? Yeah, Peter.
So I guess your model performance is also depending on the training data quality. And in mass spec-based methods, sometimes we have a noise that is, in one that is smaller than in others, so it's vary a lot. And I guess if you train your model with completely random data, it should learn nothing. And if you have extremely high quality data without noise, it should have the perfect. So something you have in between. So how which type of suggestion you can give us, to also measure the noise content for the training in different, very varied type of data?
Yeah. Yeah, so I think the main application that would be roughly similar. So we have models for example, image reconstruction from, for example, MRI images from very few case space samples, where there is then also a lot of noise in the data, or depending how you sample. So you have specific architectures that are meant, so called recurrent inference machines, for example, that are specifically designed to help deal with noisy data or undersampled data by trying to iteratively reconstruct the data and then see again if it fits or have specific noise models within the architecture. Not sure if that would immediately work for mass spec data, but there are ways to deal with lower quality data in the learning process.
Can you learn the noise from your data?
Depends a bit. So the main applications I've seen is where there is some inherent noise model that you can assume, right? So like the most simple one is, of course, there's Gaussian noise, but then you can just essentially learn the parameters of this noise model at the same time as you learn relevant information from the data. But yeah, it depends a bit on how well you would be able to model the noise, or how, yeah, I don't know that for mass.
So you need to
Okay,
have a model.
one last question before we go for coffee.
Fantastic talk. Thank you very much. I was just wondering about the sample size requirement, particularly when we talk about learning from omics data. I mean, in general, we know if you have more data points, more samples, it's always better. But on the other hand, we cannot go on collecting more and more data. So from your experience, could you say something about at what sample size there is saturation, and anything that we can learn to do omics analysis? Thanks.
Yeah, it's very hard to be very specific. I would say the easier the task, for example, if we compare it to what we did in imaging, the easier the task is for the pathologist, typically the less data you need to actually get a computer to be able to do the same. So the more complicated tasks, so if there's essentially more noise or less signal in the data, you need more samples to be able to learn that signal. What we do see over time is that actually the data requirements to train good quality models is actually getting lower. And that's, for example, caused by foundation models. So if you take a imaging foundation model as a base, and for example, specifically this, the pathology one, if you would start from scratch, it has to learn everything. It has to learn what a cell is. It has to learn how stroma looks like. If you start from a foundation model, that it already knows. So it can focus only on the things that are specific for that disease. So that's why I showed that one example. So there they could do cancer detection or segmentation with only tens of samples. And if I would do that 10 years ago, training from scratch, I would need hundreds of samples to do the same question. And I would imagine the same thing, for example, with the, well, not sure if that's relevant for the majority of the audience here, but this DNA foundation model, if you do stuff with DNA, if you start from that, then probably the amount of data you need for your specific application will be lower.
Okay, thank you.